Hey guys, we are continuing with chapter 3. Today we are working on section 3, which are lipids. Lipids are varied in structure. They're very large, non-polar molecules, meaning they share their electrons evenly throughout the entire molecule, and they are insoluble in water due to that non-polarness of them. They don't dissolve in water, they separate. They don't like to mix at all. Lipids are used in long-term storage, structural components, heat retention, cell communication and regulation, and protection. There are several different types of lipids. They are fats, oils, phospholipids, steroids, and waxes. Here's a table listing the type of lipid, the function, and the uses that humans have for them. Fats are long-term storage molecules in animals. They act as insulation as well to keep them warm in the wintertime. Human uses, butter and lard. Oils. Oils are long-term energy storage in plants. A lot of times they store that energy in their seeds, and then we use that oil for cooking. Phospholipids are a part of the plasma membrane, which we are going to talk about in great detail later on this year. We use them as food additives. Steroids are components of the plasma membrane as well. They're in the form of cholesterol, which helps hold the phospholipids together, which we're going to spend a lot more time talking about. Steroids are also found in sex hormones like estrogen and testosterone. They also form the base of a lot of medicines. And lastly, waxes. They are used for protection, prevention of water loss. Plants use them a lot to help keep water inside. Beeswax and earwax. I don't know about you guys, but I like having lips that are not chapped. Many lip balms, like chapstick, use a type of wax to help ensure that moisture is not being lost through your lips. Waxes are also used in different polishes and in candles. Long-term energy storage in the form of triglycerides. They're also called fats and oils. They function as long-term energy storage and insulation. They consist of one glycerol molecule linked to 3 fatty acids by dehydration synthesis, which you will remember is bonding molecules together by taking out a water molecule. We're going to have a diagram of it in a few slides, so just sit tight and we will get there. There are different types of triglycerides. They can be saturated or unsaturated. Unsaturated triglycerides, they have a double bond or even a triple bond between some carbon atoms inside the molecule. That means that they are not holding on to as many hydrogens as possible. That's why they are unsaturated. These tend to be liquid at room temperature. Most of these come from some kind of plant source. They can have chemical groups, which you will remember from section 1, on the same side of that backbone or on opposite sides of the double bond. Saturated fatty acids or triglycerides, they have no double bonds. They are bonded to as many hydrogens as possible, and they tend to be solid at room temperature. These generally come from animal sources like lard, which is animal fat, or butter, which comes from cream, which is an animal byproduct. Trans triglycerides, they have a double bond that actually moves around. That's why it's called trans fat, because that bond can reposition itself throughout. So here is an example of a saturated fat. We have the glycerol right here, bonded to 3 fatty acid tails or chains, and you'll notice that they are very straight and they are all holding on to as many hydrogens, which are the white dots, as possible. And down here, we have an unsaturated fat. You'll notice that it's all bent and kinked up. Well, that's because there are double bonds or even triple bonds on those fatty acid tails. That makes them not line up as straight, and it makes them less dense, which makes them liquid at room temperature. This is the glycerol molecule. This is what forms this part of the molecule that all of the fatty acid tails are bonded to. We're going to go through dehydration synthesis 3 times to bond each fatty acid to the glycerol. So we're going to end up with 3 water molecules left over. So we're going to go through it 3 times, once here, and once here. And we end up with a triglyceride. You'll notice here we have a double bond, which results in a bent or a kinked strand. Moving on to our next category, phospholipids. They are part of membranes, part of the plasma membrane. Their structure is very similar to a triglyceride. It consists of 1 glycerol molecule linked to only 2 fatty acid chains and a modified phosphate group. The fatty acid tails are nonpolar and hydrophobic, meaning they don't like water. The phosphate head or phosphate group is polar and it really likes water. Okay, it's hydrophilic. They make up the plasma membrane of our cells, and when they are put in water, they line up or aggregate to form a lipid bilayer, meaning 2 layers. The polar phosphate heads are oriented outward toward the water because they are hydrophilic. The nonpolar fatty acid tails are oriented away from the water, and they form a hydrophobic core. The kinks in the tails of those fatty acids, they keep the plasma membrane fluid across a range of temperatures. So it's important for them to have that double or triple bond, which is going to make it easier for them to tolerate temperature changes. So, sorry about that. Here's a picture. Okay, here is the polar head, the phosphate bonded to the glycerol, which is then attached to the fatty acid tails. You'll notice the double bond right here. That is right where the bend or the kink is going to occur. And this prevents them from lining up too closely together, which is important for membranes so that they can let things through when they are supposed to, which we're going to spend more time later on discussing that. Our next category, steroids. They are 4 fused carbon rings. They are composed of 4 fused carbon rings. They have various functional groups, remember from section 1, attached to that carbon skeleton. They are components of animal cell membrane regulation. They help control what goes in and out of the cell. Examples include cholesterol, testosterone, and estrogen. Hopefully you have heard those terms before. You may not know exactly what they mean, but that's fine. Testosterone and estrogen are sex hormones, and they differ only slightly in the functional group that are attached to the same carbon skeleton. Cholesterol is a precursor for lots of steroids that are made and used inside your body. So it's important that you have good cholesterol in your body. When you hear someone talk about having high cholesterol, they mean the bad kind that can contribute to circulatory disorders, which can cause a buildup of plaque molecules in your arteries that can lead to a heart attack. My dad has high cholesterol, and so he takes what's called a statin, which helps lower that bad cholesterol and helps prevent buildup in his arteries. So here we have a cholesterol molecule that is either being turned into testosterone or estrogen. And depending on which one the body has, you will develop different characteristics. We have a male peacock that has a large amount of testosterone, which signals his body to grow these big, magnificent tail feathers. Or over here we have a female who does not have the big, extravagant tail feathers. That is, her body is not producing those because it has a large amount of estrogen, which is not telling her body to make these big tail feathers. Waxes. Waxes are long chains of fatty acids connected to carbon chains containing alcohol functional groups. They are solid at room temperature. They are waterproof, meaning that they don't mix with water. They are resistant to degradation. They don't break down very easily. They function in protecting. They protect plants from water loss. In humans, we produce earwax to help protect our ear canal from things getting in there that shouldn't be there. Bees produce wax in their hives. It's where they, when the queen lays their eggs, and that is where they store their food. So here we have a bumblebee in his hive. I think that's a bumblebee. We're going to call it a bumblebee. I don't see any eggs in these in the hive, but that's okay. I'm sure he's just adding food there. Over here we have some kind of fruit. I'm not exactly sure what those are, but you'll notice the leaves are waxy, and they are helping prevent water loss. So that's it for lipids, guys. If you have any questions, go back, watch the video again, shoot me an email, or ask me in class tomorrow.